biology revision. So, we only have today, Monday, Tuesday, Wednesday, and then that's that. That is all the revision we have for paper 1. Honestly, I feel like I need more time. I really do. There's certain topics that I'm starting to forget more and more. We really are in this crunching period now where we need to just get as much remembered as possible. And what we were doing yesterday was we started looking at paper 1 for 2021. And we got up to question, I want to say 7. It was a somewhat decent paper. We lost a lot of marks. We lost quite a few marks here for some genuinely difficult questions. Then we lost a couple marks here for some pretty easy questions that I misread. So I'm just going to quickly count up these. So we got 33 correct and 13 incorrect. So I want to say that that's like 60-something%. That is out of 46, that's 33 correct. Yep. So that's probably about like 60-something% correct, which is an A. Anything above a 60 is basically going to be an A, unless you get really unfortunate grade boundaries. So, let's have a look at these new questions. We'll just quickly pull this up. 33 out of 46 is 71%. That would be an A star. Okay, so the next question. Some of the antigens found on the surface of tumour cells are also found on the surface of healthy human cells. Use this information to explain why treatment with an ADC often causes side effects. So what we're going to say, we're going to say that ADC will bind to receptor on healthy cells because receptor is the same / complementary. I think that would be a mark. And then what we need to say is ADC will release drug killing cells. So then ADC releases drug into healthy cells. And it says that the drug leads to cell death. So we're going to say cell death of healthy cells. Right, that was a very long-winded explanation, but I hope that I got that right. ADC will bind to non-tumor / healthy cells, reject reference to active site. Wait, have I over-explained this? Cause death / damage to healthy cells. Okay, so the only bit I needed to say, I didn't need to say anything about the receptor being complementary. I didn't need to say anything about the drug. I would have gotten 2 marks just for saying this. It will bind to receptor on healthy cells. Cell death causes cell death. Alright. 2 marks. Yes, I definitely over-explained, but I still would have gotten both marks. Okay. Whoa, this is a lot of information. Scientists investigated whether one type of ADC could be used to treat human breast cancer. This ADC is a monoclonal antibody combined with a drug to inhibit mitosis. Okay. The monoclonal antibody... Wait, is the figure even... Okay, the figure's not shown. We can't answer. We cannot answer that question. And we can't do the follow-up question either. There is one we could possibly do. Suggest and explain 2 further investigations that should be done before this ADC is tested on humans. So it's been tested on mice. I don't know. I don't know whether we have the information to infer. Test on other mammals, test on healthy humans to look for side effects. Investigate different concentrations to find a safe dosage. Okay, that would be a nice question then. So, if you're asked in an exam, they've done this trial for this drug. Drug trials, before testing it on unhealthy humans, test on healthy humans, allowing you to look at side effects. Test on different mammals, because mice might have a different response to, say, I don't know, like a pig or something. You want to try different dosages. I try different concentrations of the drug to find the optimal dosage. So, that would have been a doable question. Honestly, I think I just panicked because I didn't see the figure. But no, I could have done that question. Okay, next question. Describe how a triglyceride molecule is formed. 3 marks. Okay. We are going to say that it is a condensation reaction. It is the formation of fatty acid What is the structure? It's a fatty acid. It's 3 fatty acids and 1 glycerol. It's 3 fatty acids, 1 glycerol. It's the condensation of that. And do we have to give a specific location? I'm going to say in smooth endoplasmic reticulum. Let's have a look. 1 glycerol and it's running. 1 glycerol and 3 fatty acids. Oh, okay, we would have got no marks. We would have got 1 mark. Would have dropped 2. Whoa, wait, it's running a lot. Condensation reactions and removal of 3 molecules of water. You would have had to say that? No mark for mentioning where it occurs. Ester bonds form. Okay. So let's redo this. You can draw a diagram, so that's what I would ultimately do. Glycerol, 3 times fatty. It's the condensation, so it's the elimination of 3 H2O molecules. And it forms ester bonds. Shit, honestly, I knew all that information. I know condensation is the removal of H2O. I know the ester bonds form. I just didn't realise you had to mention it. That's unfortunate. Whoa, okay, what is this? Table 5 shows some properties of 4 fatty acids. Whoa, I've not seen a question like this before. Put a tick that shows the correct information. So caprylic acid, so let's look. We got caprylic, so this is number of carbon atoms in the R group and number of double bonds in the R group. So caprylic acid has 8 and 0. palmioretric, I'll just put pal, has 16 and 1. So that would be mono unsaturated, that would be saturated because no double bonds. Stearic and lin, which would be 18 and 2. So it is saying which one. Okay, firstly, this one is going to be saturated. This one's going to be saturated. That's going to be unsaturated. That's going to be unsaturated. Is that going to help us in anywhere? Caprylic acid. Linec is saturated. It's not. Caprylic is unsaturated. It's not. Palmetric is unsaturated. That's true. Stearic is saturated. That's true. So it's one of these two. Diagram M has a kinky chain, but only unsaturated have kinky chains, so it's palmetric. Yes, okay. That was a difficult question, you know. That got me thinking for quite some time there. That was about eliminating the odd one out. That was a fun question. It's holy shit, it's running so much. That's crazy. Okay. Man. This is a big question. Okay. The percentage of saturated fatty acids compared with unsaturated found in lipid stores and seeds differs in different populations. So they compared two populations of a plant. They grew young plants from seeds and collected. They planted seeds on wet tissue paper so that root growth was visible. They grew them at two temperatures, warm and cool. The scientists used a data logger to measure the length of the root rather than a ruler. Why would you use a data logger, and why is this important? What the fuck is a data logger? Literally just showing, I don't know. We're gonna have to look at this. Okay, so that's to increase accuracy because the differences are going to be small. Okay, no idea what a data logger is, but if you're asked a question about that, it's to increase accuracy. It is known that during respiration, saturated fatty acids yield more energy. So saturated yield more energy than unsaturated. Saturated have a higher melting point, so energy, melting points. And lipases in seeds act more rapidly on liquid substrates. So lipases act on liquid. So, show how each population is better adapted for its natural environment. So, population 1 is in a warm climate. The mean percentage of fatty acids that are saturated, therefore, is higher this is kind of a difficult one to explain. called its lower percentage of sat fats, higher percent obviously a greater percentage of saturated fat means a greater rate of energy, well, a greater increase of energy yield in respiration. Cold, obviously less saturated fatty acid is beneficial because then it means that unsaturated fatty acids will be more likely to be liquid, meaning that they can be used faster, greater lipase action. Lipase action as unsats fats has lower melting points. In warm, a greater amount of saturated fatty acid means that the energy yield in respiration is going to be greater, and a greater percentage of saturated fatty acid in warm conditions means that it can still become a liquid because it has a higher melting point. Let's see if we're right. Population 1 grew longer roots in warm temperatures. Standard deviation, obvious one to mention. Standard deviations do not overlap, so difference is unlikely to be from chance. Population 1 is better adapted to warm conditions because it has more saturated fatty acids, so more energy available for growth, we should have said. Population 2 is better adapted to cool conditions, so it has more unsaturated, so more lipase activity. Whoa, it's nice and sunny now, what the hell? So, we would have maybe got 2 marks for that. Again, we under-explained ourselves. This is a big problem. One is that the error bars overlapped. That was a really obvious mark point we could have said. The second one is literally stating the obvious. Population 1 grew longer roots in warm temperatures. Population 2 grew longer roots in cool temperatures. That's all you had to say. So, 1 mark point for stating the obvious, which we didn't do. 1 mark point lost because we never said that the standard deviations overlap. So, when the standard deviation... they don't overlap. So it means that the difference is likely to be... not likely. Although these 2 populations are completely separate and show variation, they are both called the same thing. Explain why. This is because they are still able to interbreed to produce fertile offspring. It can mate to produce fertile offspring because it's the same species. Okay. So again, we missed out on some very obvious marks, but we still did okay. Honestly, a lot of that information was very overwhelming. Table 7, okay. This is kind of like an Ethan level question. This is only 2 marks, which is kind of weird. Carbon, nitrogen, oxygen, and phosphorus, which I think is just a P. And we have everyone's favourite galactose, phospholipid, RNA, and sucrose. So which elements are found? Okay, well let's break this down slowly. Galactose is not going to contain nitrogen. no phosphorus. Because that is literally, it is literally just a monosaccharide. Galactose plus glucose is lactose. It's a monosaccharide. So it's going to contain carbon and oxygen. Phospholipid, on the other hand, is also going to contain carbon and oxygen, but it's also going to contain, of course, a phosphate. Now, RNA, ribose nucleic acid, obviously it contains a ribose sugar, so it's going to contain a C and an O. RNA, the structure is a base, the sugar, and of course a phosphate. The base contains nitrogen because it's a nitrogenous base. Phosphate. Sucrose is gonna be the same as galactose, I believe. So, I think that that's everything. Please be right. Yes. Okay. Nice 2 marks. Honestly, kind of wish there were more marks for that question, because I feel like a lot of people would have got one of those wrong somehow. But again, that's why it's important to know and be able to compare the structure of these different biological molecules. You know, you look at, for example, ATP. That's where you have your adenine base, your ribose sugar, and obviously your inorganic phosphates. But then we go over and look at something like DNA, and hold up, or RNA, let's just say, for the sake of convenience, and hold up, RNA also contains a nitrogenous base, it also contains a ribose sugar, and it also contains an inorganic phosphate. So you could easily get asked questions in an exam where you're having to compare and contrast the structures of certain biological molecules, and you could really get thrown off guard if you don't know them well enough. Oh, what is this question? Oh no, this is with a semi-conservative replication. After Watson and Crick proposed the model, scientists investigated the possible mechanisms. They grew a bacterial population, providing them with a nitrogen source containing only the heavy isotope. So heavy is N15. As soon as all the DNA contained this heavy nitrogen, they changed it to light nitrogen. N14. And they changed it at 0 hours. The generation time for a population of bacteria is the time taken for all the bacteria to divide once by binary fission. Use figure 11 and the following equation to calculate the generation time for this population of bacteria. Give your answer in hours. What the fuck? Okay. Right. Log 10 followed by the size of a population at plus 4 hours divided by the population time at minus 4 divided by log 10 to the power of 2. No idea what that means in the slightest, but the size of a population at minus 4 is... Whoa. Why is the graph looking like this? Whoa, why is the graph looking like this? Is this an error, or is this how the graph's formatted? I have zero idea how you're meant to infer this graph. For context, it's like the grid lines are like... It's like the grid lines do that instead of going up evenly. I have no idea how you're meant to interpret this graph. We've never done anything like this before. Should I say 10 to the power of... I don't know. I'm going to have to look at the answer. Evidence of 4 times 10 to 6 and 3 times 10 to... Okay, let's look. 3 times 10... 1, 2... Honestly, this is an odd question. I don't know whether the graph is wrong or whether it literally is just completely... I wouldn't know how to interpret this graph in the slightest. It says at -4 hours, it's up by 1, 2, it's up by 3. So is that going to imply 3 to the power of 6? No, 1, 2, 3. That would imply 3 to the power of 6. Sorry, 3 times 10 to the power of 6, but it says 4. And here, at 4, I would have read this as 2 times 10 to the power of 9, but it's 3 times 10 to the power of 9. So I'm assuming the way you're meant to interpret that graph and the grid lines is, I think it's meant to be logarithmic. I'm assuming that's what you're meant to do, but we haven't done anything like this ever. I don't know how you are meant to interpret that. Because then obviously I'm assuming that if it says something like 2, then somehow it's actually a 3. Which is where you would have got it from. So honestly, I think I could have answered that now. Would not have been able to answer that though in an exam, because we have not been taught that. Anyways, the next question. At intervals, they remove samples of bacterial population. DNA made using N15 has a higher density. So, at generation 0, we'll just have to draw it like this. At generation 0, generation 1, 2, and 3. At 3, it is like this. So it's very dense. Generation 1, it's kind of in the middle. Generation 2, it's like that. Generation 3, it's like that. So, which of these models, P, Q, or R, is supported by the results? Give the letter and name of the model supported, and explain why the results do not support the others. So I think it's probably model Q, because it is showing the first generation it's heavy, second generation it's 50-50, third generation it's 75-25, generation after that it's etc, etc. So it goes from being like, oh, and I'm gonna... Sorry about that. I'm assuming it's model Q. I just want to check if I'm right there. Model Q, and yes, it is semi-conservative. How many marks would that be? 1. Now we need to give reasons why the other ones don't. Why would model P not work? Explain why the results do not support the other models. It wouldn't support P because then it wouldn't support it because by the second generation, by the second generation, it would show what generation 3 would be. So I guess your explanation there would, the second generation, there would be a high bar and a small one when there isn't. For R, your justification would be that the graph is not even. Model P, because there should be 2 peaks in generation 1. Wait, really? Yeah, or there should be a 3-to-1 ratio of peaks in generation 2. So basically what I said, obviously I wouldn't have gotten a mark because I didn't explain it like that, but Model R is unsupported because there should be greater than 2 peaks in generation 2 and 3. Wait, how would that justify it? Oh, because there's... Okay, so the... Oh, I didn't see. So R isn't even. Each one has a different density. So there would be more peaks if it was R, and there would be a greater peak like that in a 3-to-1 ratio. So I'm going to give myself 1 mark, give myself 1 wrong. In an exam, I likely could have explained how Model P worked. I definitely could have said that there should be 2 peaks in generation 1. Because, yeah, you have one that's light, one that's heavy, instead of it being even. So there'd be 2 peaks. Honestly, that was kind of a creative question. I'll give them that. But please, can we be done with this now? Yes, okay. Describe the structure of DNA. This is a 5-mark question, so we are once again going to strive for 6 points, but let's just say 5. DNA, right. A. That's the base. Ribose sugar. Sorry, deoxyribose sugar. And our phosphate ion. So, that is what we are going to specify, firstly. It is nitrogenous base, inorganic phosphate ion, and deoxyribose. So those are its structural components. Next we'll talk about the bonding. We'll say phosphodiester bonds join adjacent nucleotides, nitrogenous base, and this forms the sugar phosphate backbone. And we are going to say that hydrogen bonds join together complementary bases. Now it's saying describe a structure, so we should probably specify the idea that the hydrogen bonds are weak, which allows the DNA molecule to easily be unzipped for replication / transcription. And we should easily specify that the adenine base, I guess you could say the sugar phosphate backbone provides strong structural supports. And also we should specify that the bases are used to hold information, and that they have complementary base pairing. So that's adenine to thymine, thymine to adenine, cytosine to guanine, guanine to cytosine. Let's see how close we were. So it's a polymer of nucleotides. Wouldn't have gotten a mark for that. Great, so already lost a mark. Each nucleotide is formed from deoxyribose, a phosphate group, and a nitrogenous base. Okay, 1 mark. 1 mark for saying all of that. Phosphodiester bonds between adjacent nucleotides. Yes, 1 mark. Double helix / 2 strands held together by hydrogen bonds. Hydrogen bonds / pairing between adenine, thymine, and cytosine, guanine. Really? I think that is a very unfair question. Number 1, I got the mark scheme confused. Again, this is the problem with biology. This is why I hate AQA. They make you think in terms of mark schemes. They don't make you think in terms of biology. So what I was doing there was I was remembering a time where there was a similar question where they asked you to describe how the structure is important for its function. I would have gotten all of these marks if it was that question. Unfortunately, I only got these 2 marks. I should have said that it's a polymer of nucleotides. Then I should have elaborated what each nucleotide is made of. I should have then gone on to say that the double helix is held together by hydrogen bonds because the hydrogen bonds complementary base pairing. You know what? I'm going to give myself a mark because I did talk about the base pairs, and in an exam now, I would have known to talk specifically about how it's hydrogen bonds. Hydrogen bonds join together complementary bases. I would have definitely then said A to T, T to A, etc. So yeah, that was a difficult question, purely because I misread it, but next question. Name and describe 5 ways substances can move across the cell surface membrane into a cell. Okay, number 1, simple diffusion. Number 2, facilitated diffusion. Number 3, active transport. Number 4, co-transport. Number 5, osmosis. Just rattle them off. Easy as. Okay, simple diffusion. That is the movement of non-polar, lipid soluble and small molecules across the cell surface membrane down a concentration gradient. And it's a passive process. 2, facilitated diffusion. That is the movement of polar molecules, such as ions, using a transport protein, and again it's going to be down a concentration gradient, so it's passive. Then we move on to active transport. This is the movement of ions. The movement of ions against concentration gradients using a carrier protein, and it's going to be using ATP. Co-transport involves a symporter, and it is the movement of 2 separate substances. I don't know what else you would say there. Definitely the idea that it involves facilitated diffusion and active transport. And finally, osmosis is once again, it is a passive process. It is moving down a water potential gradient. Osmosis is in a selectively permeable membrane. And again, it's the movement of water molecules down a water potential gradient through or across a selectively permeable membrane. Let's have a look. Simple diffusion of small nonpolar molecules down a concentration gradient. Correct. Facilitated diffusion down a concentration gradient via a protein carrier/channel. Yes, so we'll just say there a carrier/channel. So yes, 1 mark. Osmosis of water down a water potential gradient. I said down a water potential gradient through, yes, 1 mark. Active transport against concentration gradient via protein carrier using ATP. Co-transport, 2 different substances using a symporter. And we don't need to mention that. So, 5 marks. Whoa, there's another question. For 5 marks? What the fuck? I've never seen it where it's been 3. I thought the paper was over. There's more. Compare and contrast the structure of the 2 cells visible in the electron micrograph. It's showing 1 animal cell, 1 prokaryote. Oh, okay. Now, what does it want us to say specifically? The structure of the 2 cells visible. So, we are going to mention that the animal cell is going to have a cell surface membrane. The prokaryote is going to have a cell wall. Is also going to have a cell wall made of murine. And it's also going to have a slime capsule. Then we should talk about the idea that in the animal cell it has linear... Wait, no, would DNA be a thing we talk about? It's saying contrast the structure of the 2 cells visible. Contrast the structure of the 2 cells visible. I guess you know what, we'll just say animal cells have linear DNA, prokaryotes have circular. Doesn't matter if that's worth a mark, it's just important to write that down. Next, we will say that in animal cells, they have, is it 80S, 70S ribosomes? Prokaryotes, they have, no, it's 80S and 70S ribosomes. Animal cells contain 80S ribosomes, 70S in the mitochondria. Prokaryotes only contain 70S ribosomes. I guess also the fact that prokaryotes have no membrane-bound organelles, whereas these do. Let's have a look at what the mark scheme says. A has a nuclei, whereas B, oh my god, no, these would be so easy to get marks out of. Really? Hold on. Except in all marking points. A has linear DNA, whereas B has circular DNA. Yes, 1 mark. A has no cell wall, whereas B has a murine cell wall. Yes, but I could have divided this into 2 mark points. Would they be... I don't know how that would work. If I mentioned the marking point... That's an odd one. If I mentioned 2 marking points in the same point, would I get a mark? Because there I said the cell A only has a cell surface membrane, whereas B has a murine cell wall and a slime capsule. I guess probably not, because I haven't been clear enough. If I wanted to get 2 marks, I should have said A has no cell wall, A has no slime capsule. I'll give myself a mark, although realistically I deserved both. A has larger ribosomes. I guess that's probably a mark. I said 8ES, 7ES. Where is the mark? Really? There's no mark for saying no membrane-bound organelles. That's pretty disappointing, actually. I would have assumed that would have been the easiest mark point to get, but no. Okay. So, obviously I didn't answer that properly. I was just writing very fast notes. That would have been a piss easy question. That would have been a real easy question to milk marks out of. Literally saying A has a nucleus, B does not. A has rough endoplasmic reticulum, B does not. Literally giving away marks at that point. How the fuck does this paper have such nice grey boundaries? Like, this is one of the easiest papers ever. And somehow I've still flopped. That's wild. Okay. I'm now going to count up how many marks I actually got for this whole paper. 2, 4, 6, 7, 9. 9 incorrect. 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 21 correct. So, 33 plus 21. What the fuck am I doing? Hold on. We just said that that's 30 total. So that's going to be 21 plus 33 out of 46 plus 30. All in all, I got 71% for this paper. Pretty decent, to be honest. Obviously, there were some questions that I couldn't answer because the figures just weren't there, so I couldn't answer them properly. I remember I removed 2 marks because I've already answered one set of those questions before. So all in all, about 70%. I'm happy with that. I'm actually pretty happy with 70%. That is a good sign, because bear in mind, like, some of those questions were pretty abysmal. Like, yeah, we got basically, 11 marks for free because of that question about describing the structure of a nuclei, but I could have done better on even those questions. So, all in all, I'm wrapping things up there today. I've done like 3 hours of revision at this point. I will be back tomorrow, probably Tuesday, and definitely Wednesday. We'll go over more past papers. We'll get this hammered in. We are really, short for time now, so I want to try and do as much as possible before paper 1. And yeah, 70% isn't that bad. I'm happy with that. That would obviously be an A star on average, depending on, the boundaries. obviously, if we got a paper like that and the boundaries were low, that would be an easy A star. But yeah, that seems to be a good sign of things to come. Hopefully, I can just revise those couple extra things that I missed. Like, you know, being able to describe the structure of a DNA. I know how to do that, and yet I dropped a lot of marks on that question because I over-explained. I do that a lot in biology. I'll over-explain something obvious to the point where it doesn't give me a mark, which is a bit dumb, but yeah. We are nearly done, guys. We're nearly done. So that is all that I'm gonna give myself time for. Probably gonna go make like a main channel video now or something. I'll see